Hi there. This video is to support the topic kidney and homeostasis at A2 Module 4. I'm going to try and go through the topic fairly quickly, try and go through most of the content, maybe all of the content, and to cover the most relevant questions that are asked on this topic. Now, to be able to get maximum advantage from this video, you should have access to my notes and a question booklet and a set of answers for the question booklet. And the reason is that I've been making constant reference to my notes, and essentially everything that I do say during this video, every area that I cover, should be covered in the notes. I'm just maybe clarifying it for you. So it's quite a long topic, and there's quite a lot of areas to look at, and there's a lot of different types of questions. So I'm going to move fairly quickly. I'll try and move more quickly through the areas that are well understood by students and slow down a bit and maybe go into more detail with the more difficult parts. So I'm going to move fairly fast. As I say, I'm going to make constant reference to my notes here, and I'm going to use them as a guide so that I don't miss any aspects of the topic. So this topic is often referred to as homeostasis because you could be asked a definition then of homeostasis, which is the maintenance of a constant internal environment in the body. And then essentially we're looking at the role of the kidney in homeostasis. How's the kidney involved in maintaining constancy within the body? And essentially the kidneys are involved in controlling the water content of the body. The term used there is osmoregulation. So osmoregulation is the homeostatic aspect of the role of the kidney. The kidneys are also involved in getting rid of waste products. Two waste products you come across are urea and creatine. You could get asked then where urea comes from. Urea is a product of the breakdown of excess amino acids which are present in the body. When amino acids are broken down and used in respiration, the NH2 group is removed, and the NH2 is involved in a reaction there where it's converted into urea, and urea contains essentially the NH2 group. The formula for urea is carbon, oxygen, NH2 twice, so that's the formula of urea. So urea contains the NH2 group that comes from the amino acid. Now, to understand the kidney and the function of the kidney, you need to understand some stuff about water potential and solute potential. So going back to AS, you need to know that if you, for example, drink a lot of water, that water goes into your bloodstream eventually, and that causes the water potential of your bloodstream to increase. You could say it causes the solute potential of your blood to increase, move towards zero. Whereas if you don't drink much water and say do a lot of exercise or don't drink a lot of water for a period of time or take on board a lot of solutes, those solutes eventually get into your blood and they cause the water potential of your blood to decrease or become more negative. So you need to understand those terms. Essentially, the kidneys are involved in maintaining the constancy of the water potential or the solute potential of the bloodstream. So moving on quickly then, how does the kidney do this? Well, the kidney is involved in essentially what happens inside the kidney are two processes, ultrafiltration and reabsorption. Ultrafiltration. So the kidney effectively acts as a filter. Essentially what happens is as blood passes through the kidneys, the blood is under very high pressure. Because it's under high pressure, it can leak out of the blood vessels. And the blood vessels inside in the kidney, blood vessels involved are capillaries. And so the capillaries are grouped together in a little knot of capillaries inside, and that's referred to as the glomerulus, and that's contained inside a cup-like structure called the Bowman's capsule. I'm referring here to page 3 of the notes. I'm just looking at the detail then of the functional unit of the kidney, which is a nephron. So the nephron is where ultrafiltration and reabsorption takes place. Useful to know at the top of page 3 there, indicating where exactly the nephrons are located in the kidney. First of all, there are millions of nephrons in each kidney. Where exactly are they located? Well, to understand this, then you need to know the structure of the kidney, and you'll see there in question 1, it asks you to identify different regions of the kidney, region A, region C, and so on and so forth. Well, region A is called the cortex, the outer region of the kidney, and region C is the medulla, and the region in at the very center there, that is the pelvis. Now, where exactly are the nephrons located? Well, if you look on page 3 there at the top, you'll see that the nephrons are located on the boundary between the cortex and the medulla. So along the boundary between the cortex and the medulla, you've got nephrons located, such that some parts of the nephron are in the cortex region and some parts are in the medulla region. The regions of the nephron that are in the cortex region are the Bowman's capsule and the proximal convoluted tubule. What regions are in the medulla region? They extend down into the medulla region. That would be the loop of Henle and the collecting duct. So sometimes they like to ask you just where these regions are located. And you can see that clearly at the top page 3. Top page 3. And of course, you need to know the other, and you need to know the other features of the kidney, the other things like renal artery, renal vein, etc., etc. Now, looking there at the nephron then on page 3, and we're looking first of all at the process of ultrafiltration. Ultrafiltration is essentially the process by which the blood is put under pressure in the nephron, and that forces some of the blood content out of the capillaries in the glomerulus, and the content that gets forced out of the blood is essentially the plasma content, not the red blood cells, not the white blood cells, not the large proteins, a few small proteins, but it's essentially the plasma content of the blood, which is essentially the water content of the blood. And dissolved in that water, you have glucose. Dissolved in that plasma content, you have glucose, you have amino acids, you have lots of other food molecules, you have ions, you have ions such as sodium and chloride and potassium and so on, and they're all dissolved in the plasma content. And you could regard those substances as being useful substances: water, glucose, amino acids, ions, etc. But something that's also dissolved in the water, in the plasma, and the water content in the plasma is urea. So it gets forced through the filter by the high pressure that's created in the kidneys. Now, so we need then to look at the structure of the nephron, and you can see there that the nephron contains a structure called the glomerulus, and the glomerulus is really a little knot of capillaries, and it's in the glomerulus that the ultrafiltration process takes place. Ultrafiltration, that term was asked about recently. Ultrafiltration is the filtration of small molecules that are present in the plasma through a filter at high pressure. And you have to say that, get the idea of it being at high pressure. Ultrafiltration. And you can see there that the blood vessel leading to the glomerulus is referred to as the afferent vessel, sometimes also called the afferent arteriole, and the blood vessel leading away from the glomerulus is the efferent vessel or the efferent arteriole. And you'll notice that the afferent vessel is wider and the efferent vessel is narrower, and this is part of the production of part of the process that produces a high pressure blood inside the glomerulus. Also important to note there is that the efferent vessel, once it leaves the glomerulus, goes away down and wraps itself around the Bowman's capsule, wraps itself around the proximal convoluted tubule, the loop of Henle, distal convoluted tubule, etc. And that wrapping of blood vessels down in that region is generally referred to as the vasa recta. Now, a good question: what's the function of that? All those blood vessels that are wrapping around the Bowman's, wrapping around the proximal convoluted tubule, etc. And the answer is this second aspect of the process is selective reabsorption. And selective reabsorption simply means that all the good substances that were pushed through the filter in the plasma, that's glucose, amino acids, a lot of water, ions, etc., those so-called good substances or useful substances, they have to be reabsorbed from the filtrate. The filtrate being what was pushed through the filter. So those substances have to be reabsorbed. So those blood vessels in the vasa recta are wrapped around the proximal convoluted tubule and the loop of Henle, etc., in an attempt to try and reabsorb the useful substances. Now, I'm moving fairly quickly here now. So essentially, how does the nephron work? It's ultrafiltration, first of all, followed by reabsorption. And ultrafiltration, as I said, you need a definition for that. Selective reabsorption, maybe ask for a definition of that. We're going to look at the ultrafiltration first of all. And first thing we're going to look at is how do you get the high pressures created in at the glomerulus? How do you get the high pressures? Essentially, there could be three marks for that. First of all, you say that the blood arriving at the glomerulus has only travelled a short distance from the heart, and therefore it's already at high pressure. So it's at high pressure to begin with. So the blood arriving at the kidney is already at high pressure. And then the pressure is further increased by having a situation where the afferent vessel that leads into the glomerulus is wider than the blood vessel leading out, which is the efferent vessel. And because the vessel coming in is wider, it means the blood can easily get into the glomerulus, but it's restricted. Its escape from the glomerulus is slowed down. And in essence, that creates a high pressure, or increases the pressure. The third thing is the coiling of the glomerular capillaries, which creates a lot of friction and resistance to blood flow, and that further increases the pressure. So those three things create a very high pressure. High pressure of the blood inside the nephron, and that high pressure then forces the plasma content, essentially the liquid content of the blood, through the filter. And only molecules that are small enough can get through the filter. Large molecules such as red blood cells, white blood cells, large proteins, some small proteins, etc., they can't get through the filter. But a few small proteins can get through the filter, and this is important. You could get a question based on this, a few small proteins to get through the filter. So next thing I want to look at, well, before we look at let's have a look just at the filter first of all. Students get very worried about the structure of the filter, but really there's not a tremendous lot that gets asked about it. First of all, you need to know that it consists of three components. First of all, as the blood passes through the glomerulus, it's traveling through capillaries, and those capillaries have walls made one cell thick of squamous endothelial cells. But between the cells there are holes or pores. So if you have blood at high pressure traveling through those capillaries, those pores let the plasma leak out. So that's the first aspect of the filter. So blood plasma can leak out through the pores in the capillaries. Wrapped around the capillaries then is something called the basement membrane, which is a kind of a fibrous tissue type layer, a bit like paper in structure with lots of fibers with very small pores in it. And it's considered to be the fine filter, the aspect of the filter that really decides what does get through. And it's it that stops the large proteins. It lets some small proteins through. And of course it lets through water from the plasma with dissolved ions and glucose and amino acids and so on. But it's it that's considered to be the effective filter. And often they'll ask you, what is the effective filter? What is it that really decides what gets through the filter? And it's the basement membrane. Third aspect is the podocyte cells, and they look difficult and awkward to understand how they work, but they tend not to be so significant with regards to questions that you get asked. It's sufficient to know that these podocyte cells have like extensions or fingers, which create what are called filtration slits. So there's like gaps between these filtration slits that allow molecules to get through. But the gaps are very big, so really in effect the podocyte cells don't seem to have a useful function there in the sense that they don't really do much filtering, because the basement membrane is going to decide what actually does get through. Now, if you're given an essay question on this, they need you to list the three components of the filter there. So you need to be able to say there's the capillaries, and then the basement membrane, and the podocyte cells. And you have to give them in the correct order, in the order in which the filtrate passes through them, or the plasma passes through them. So you have to know that it's the capillaries first, then the basement membrane, and then surrounded by the podocyte cells. Note also on the diagram there at the bottom of page 6, the podocyte cells are actually located in the wall of the Bowman's capsule. So that's their location, the position in the wall of the Bowman's capsule, and that's the filter. And page 7 then I discussed the details of the filter. So the capillary pores and the podocyte filtration slits are both large, allowing all sizes of molecule to pass through, but it's the basement membrane which actually determines what does get through. It's a very fine filter. It's the basement membrane that stops the big protein. So I've just considered there on page 7 what things do get through and what does get through. I've said no red blood cells, no white blood cells, no large proteins, they're all too big. Some small blood proteins. What does get through is water with dissolved glucose and amino acids, urea, sodium, and chloride ions all pass through the filter because everything there is small enough. Okay, now as I go along, I'll deal with some of the questions. I think maybe the first aspect I'm going to deal with is if you turn to page 17 in the notes there, you'll see there's a discussion about the idea of water potential with regards to the filtration process. And here you have to consider that effectively the main part of the filtration process is water. The main substance going through the filter is water. And you can apply the ideas of water potential to the movement of water through the filter. And in essence, what you can do is you can determine the water potential of the blood plasma on one side of the filter, and you can determine the water potential of the filtrate that's gone through the filter on the other side. And for the water to move through the filter, essentially you need the water potential on the blood side to be higher than the water potential on the filtrate side. So you need a higher water potential. So the blood goes from a higher water potential to lower water potential. Now, when you're considering how you would calculate the water potential on either side of the filter, you can use the equation that you remember from AS. The water potential, given that sum, is equal to the solute potential plus the pressure potential. Yep, and we can think about all those terms. So if we're considering the water potential of the blood on one side of the filter, we can say that's a combination of the solute potential and the pressure potential. Well, the pressure potential in that case would refer to the high pressure created, the high hydrostatic pressure created by the pumping action of the heart and the constriction of the afferent arteriole and so on and so forth. So the high pressure, so that term there it relates to the high pressure created at the filter. And if you look on page 17, then I've given an illustration of this. I said if you look at the blood plasma, I said in that particular example there, I said the pressure potential is +6, and this is due to the high pressure of the blood entering the glomerulus. But counteracting that, is there anything resisting? The high pressure is trying to force the plasma through the filter. Something resists that, and the thing that resists it is the solute potential of the plasma. And the solute potential, you see in that case there, so the solute potential is -3.3. In other words, it's going to resist the pressure potential. And the solute potential is largely due to the solute content of the plasma, which would be mostly it would appear to be due to the large proteins. So when you take those two values, +6 for the pressure potential created by the pumping action of the heart, and the -3.3, you'll see in my example there, you work out the water potential of the plasma is +2.7. So it's a positive value. So a high water potential of the plasma. Now, what else do we have to consider? Well, we have to go to the filtrate and consider the solute potential of the filtrate and the pressure potential of the filtrate. That is, if we're going to calculate the water potential of the filtrate. And you'll see in this case, the solute potential of the filtrate is quite a small value. It's -0.3, and it could be due to the few small proteins that actually get through in the filtrate. It's mostly due to those small proteins. It's also due to some of the glucose and amino acids and so on that lowers the water potential, lowers the solute potential there. But there's also some back pressure from the filtrate. In other words, the build-up of filtrate when it goes through the filter creates a back pressure, and that gives you that pressure potential value of +1.2. When you add those two values together, you get a water potential of the filtrate of +0.9. So from that illustration, you can see the water potential of the plasma is +2.7. That's very high, and the water potential of the filtrate is +0.9. And so you can see one water potential is much higher, and the other water potential of the plasma is much higher, and that means in essence water is going to move from the plasma across the filter into the filtrate. The difference between the water potential in the plasma and the water potential in the filtrate is given the term the net filtration rate. net filtration rate. And from my illustration now, the net filtration rate is simply you just subtract one of those two values, one from the other. So you subtract 0.9 from 2.7, and you get a value of 1.8 kilopascals. And the greater the difference, the greater this net filtration force, the faster the rate of filtration. Now, a question on this has never ever come up except on one occasion, and that was just looking at and it's not in my question book, but if you want to seek out this question, it's January 13's paper, January 13, and it's question 2. And there's a question about this. It didn't really go into much detail on it. If you look at that question, you'll see the second part of that question asks you to do a similar calculation as I've done on the table there. And they've asked you to work out the net filtration force in that example. And I think in that example, the answer is plus 2.3 is the net filtration force in that particular question. So I deliberately left this towards the end of the notes because this has never come up in any other questions since 2013, which is a long time ago, which is almost 10 years ago. So it's don't get panicky about this bit. It's really quite a lesser component. You might come across it in a question, but essentially that's what it involves calculating the water potential size through the filtrate and discovering that the water potential on the blood side is higher than the water potential on the filtrate side, and therefore that causes water to move from the blood side to the filtrate side down the water potential gradient. And the difference between the two sides refer to the net filtration force. So that's that question. Once again, January 13, question 2, and you can look through that question. Just looking at the end of that question there, that January 13 question, it says that people with high blood pressure may find that there's protein in their urine. High blood pressure. Well, the argument there is with normal blood pressure, the pressure isn't great enough to push the large proteins through the filter. But if you suffer from high blood pressure, it'll push small proteins through, but also push some of the large proteins through. Now, the proteins do get reabsorbed in the proximal convoluted tubule. But if you've got high blood pressure, you're pushing too much protein through the filter, and there's not sufficient time, as it were, for it to all get reabsorbed. So some of the protein that gets pushed through the filter doesn't get reabsorbed, and it ends up in the urine. And so this is an indication. Doctors can test the urine of patient. If they discover protein in the urine, then they're saying that's a suggestion that there's high blood pressure in this patient, or there's some damage to the filter. So the answer to that question is, what happens? You have high blood pressure, you have protein in your urine. Why is the protein in your urine? So the high blood pressure pushes extra proteins through the filter, and those extra proteins don't get reabsorbed in the proximal convoluted tubule. Normally the small proteins that do get pushed through the filter get reabsorbed by pinocytosis. Okay, so that's that aspect dealt with. I now want to move on to the idea of selective reabsorption. So selective reabsorption is essentially so far the kidney has pushed most of the liquid part of the blood through the filter. That's the plasma containing all the good things and the urea as well. Now, selective reabsorption takes place throughout the rest of the nephron. So we're talking proximal convoluted tubule, lipophenol, and distal convoluted tubule, and collecting duct. Although the distal convoluted tubule is not on the course, so don't worry about anything about it. On top page 8 there, it said selective reabsorption. It said the main substances which need to be reabsorbed, you need to reabsorb glucose, amino acids, sodium and chloride ions, and water. These are the main substances that you look at being reabsorbed. And just I've listed there where exactly they do get reabsorbed. All the glucose is reabsorbed in the proximal convoluted tubule. All the amino acids get reabsorbed in the proximal convoluted tubule. Sodium and chloride ions get reabsorbed in the proximal convoluted tubule and the lipophenol, and water gets reabsorbed basically everywhere. Proximal convoluted tubule, lipophenol, and collecting duct. So first of all, reabsorption in the proximal convoluted tubule. First of all, the proximal most of the reabsorption takes place in the proximal convoluted tubule. So by the time the filtrate has made its way down along the coiled our first convoluted tubule, proximal convoluted tubule. Nearly all the good stuff has been reabsorbed. It's very effective at getting reabsorbed. And I've seen questions where they've asked, why is the tubule convoluted? Well, convoluted just means it can be longer, and therefore it's got more surface area, more time, as it were, for reabsorption, so as to ensure that all the reabsorption takes place in proximal convoluted tubule. The most frequent question with the proximal convoluted tubule would be that question there that you have on the most typical question would be the question, say for example, question 9 part B, where it shows you the cells that line the proximal convoluted tubule, which are called cuboidal epithelial cells. And it will ask you what are the main features of those, or what are the main adaptations of those cells for maximum rate of reabsorption. And the answer is those cells are very distinct by having microvilli. So you can see the microvilli at the top there, which are basically foldings of the cells. The cells are notable by having very big nucleuses. And also the other feature, they have a lot of mitochondria. So those are the two main features: the presence of microvilli to increase the surface area for reabsorption, and the presence of lots of mitochondria in those cells. Don't just say they've got mitochondria, say they have lots of mitochondria, because all cells have mitochondria. They have lots of mitochondria, and anywhere in biology where you see lots of mitochondria, you're thinking active transport. Lots of mitochondria for more respiration, for more energy released, for more active transport, because the reabsorption of glucose and amino acids and lots of ions is brought about by active transport. There's also facilitated diffusion takes place here, but active transport is the one that tends to be concentrated on in exam questions. Those cells help to reabsorb glucose and amino acids and so on. So top of page 9, I'm not spending too much time on that question, but that question comes up a lot. Give the features. This question here, question 9b part 2, just says give 2 distinct ways in which the cells of the proximal convoluted tubule are adapted for the function of selective reabsorption. And there you go. Now, top page 9, and then I've talked just about what gets reabsorbed. I said glucose and amino acids are reabsorbed by active transport. You could also include facilitated diffusion there. Sodium chloride ions are reabsorbed by active transport and diffusion. Water is reabsorbed by osmosis, and small proteins are reabsorbed by penocytosis. And that essentially is what's happening in the proximal convoluted tubule. Any substance that do get reabsorbed from the filtrate through the tubule walls goes back into the blood of the vasa recta, into those little capillaries that wrap around the proximal convoluted tubule. And that way, if they go back into that blood stays in the body then, it stays in the kidney. And therefore, whatever gets reabsorbed into those blood vessels stays in the body and doesn't go out in the filtrate. Now, I've put a question there just in the notes. I just said, why is water reabsorbed by osmosis? Well, you'll see there in the diagram at the bottom of page 8 that the glucose and amino acids and so on gets reabsorbed by active transport. Well, if you move the glucose and amino acids out of the filtrate, that raises the water potential of the filtrate. And if you move the glucose and amino acids across into the bloodstream, that lowers the water potential of the bloodstream. So effectively, you've created a water potential gradient between the contents of the filtrate and the contents of the capillaries. And therefore, water follows after the glucose and amino acids. So this is standard in biology. You get substances like ions and glucose and amino acids and such moved by active transport involving energy. And then once it's moved, then the water follows after that, because by the movement of the glucose and amino acids, you've created a water potential gradient. So water gets reabsorbed by osmosis. Okay, so moving on then to just some other discussions. There are some more difficult questions sometimes that ask you things like, why might at the top of page 10 there, you'll see I've asked a question about why might a higher temperature speed up the rate of reabsorption? Well, higher temperature in the kidneys would cause a faster rate of respiration in the cuboidal epithelial cells, and that would produce energy more quickly. And if you get energy produced more quickly, you can do active transport much more quickly. And if you can do active transport much more quickly, then you can move the amino acids and the proteins faster across the cells. The active transport carrier proteins, which you came across at AS, they work faster then because there's more energy available because it's higher temperature. Protein does not normally appear in the urine, at the bottom of page 10 there, except individuals with high blood pressure may have protein in their urine. So already dealt with that. Already dealt with that question. Now, okay, before I come to the lipid panel, I'm just going to do a quick scan of the questions here and just see which questions we need to talk about at this point. I'm going to come back to question 1, part B in a moment or two. Question 2 is okay. It just asks what's the function of the different parts of A, B, and C of the kidney. You can see that's I've outlined that on the notes there on page 18, where I've set out the precise function of each of the different parts. And then it's asking about how the glomerulus is adapted. So we've already talked about that, and so on. The rest of that question is quite difficult. I'll try and come back to that part C and part D of that question too later on. I'm going to come to question 3 in a moment or two. Question 6. Question 6. I'm going to have a quick look at question 6 there. Name the effective filter during ultrafiltration. Well, that's the basement membrane. And then it shows, it says here, the relative concentration of a range of substances found in the filtrate and the plasma can be compared. The relative concentration is expressed as something called filtration ratio. What we see for glucose is the filtration ratio is 1. What that simply means is the concentration of glucose in the blood plasma in the glomerulus is the same as the concentration of glucose in the filtrate. In other words, they have the same concentrations. And the reason why that is that at the filter, during ultrafiltration, water and glucose both get pushed through the filter. Both of them go through the filter, and they go through the filter in the same proportion as they existed in the blood in the first place. So, for example, if the blood contains, say, 1 gram of glucose per 100 mils of plasma, then the glucose gets pushed through the filter, and the water gets pushed through the filter, such that on the others in the filtrate you have essentially 1 gram of glucose per 100 mils of water. So the two get pushed through the filter in the same proportions, and that's why the ratio is the same. The same with amino acids. Amino acids and water both go through the filter, such that the concentration of amino acids in the blood plasma on one side of the filter is the same as the concentration of amino acids in the blood plasma, in the filtrate on the other side of the filter. However, small proteins, you'll see the concentration of small proteins is very tiny in the filtrate compared to in the bloodstream. It's only a fraction, and that's basically telling you that some small proteins get through, but very, very little. So water gets pushed through the filter, but the small proteins that are dissolved in the plasma don't get pushed through. So very tiny. Medium-sized proteins is even smaller. Means very few medium-sized proteins get through, and a very tiny amount of medium. And notice that urea is 1. That simply means that urea gets pushed easily through the filter because it's small enough. It goes through the filter. So essentially, in the answer scheme to that question, you'd be saying that glucose and amino acids and urea are all small molecules, and they get pushed through the filter, and they can pass through the filter easily, and they pass through the filter in the same proportion as they existed in the blood plasma in the first place. Whereas some small proteins get through the filter because they're small enough, but medium-sized proteins, very, 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 very tiny amount of them gets through the filter because they're too large, essentially. And you can check out the answer to that question. Check out the detailed answer to that question yourself. Now, I don't believe that I'm going to be able to look at all the questions here, but I'm going to quickly do the lipid panel and then come back and look at several questions that do with the lipid panel. So the lipid panel gets students very worried. It looks complicated, but it's actually quite straightforward. You'll see just in my notes there, I've just coloured it in a wee bit, just to show you the filtrate coming down the descending limb and then up the ascending limb, and then across into the collecting duct. Okay, a wee bit of colour helps you something. Okay, you'll notice there then that I've said that the lipid panel extends down into the medullarities. Okay, so that's where it's located in the kidney. And I've said essentially sodium and chloride ions and water are reabsorbed in the lipid panel. A lot of water gets reabsorbed. Now, it's actually quite straightforward with the lipid panel. Essentially, there's 3 marks to it. 3 marks to it. But the starting point, you have to start off by identifying different regions of the lip of Henle. And you say that there's a descending limb. So that's where the filtrate's going down. And then there's an ascending limb where the filtrate's come back up again. Okay, and what you see is the walls of the descending limb are permeable to water. In other words, water could leak out through the walls of the descending limb. But you say that the walls of the ascending limb are impermeable to water. So water can't leak out through the walls of the ascending limb. Now, here's the 3 marks. Where do we start? Well, we start off, first of all, we go to the ascending limb, first of all. And we say this. As the filtrate moves up the ascending limb, sodium ions and chloride ions are pumped out of the filtrate, out through the walls by active transport. So that's the first mark. In the ascending limb, you go to the ascending limb first of all, you say as filtrate moves up the ascending limb, sodium and chloride ions are pumped out, actively pumped out through the walls, through the cells and the walls by active transport. So there's the sodium and chloride ions coming out. And once they're out of the filtrate, they're now outside the lip of Henle. They're now out in this region. And this region is often referred to as the medulla region. So you say the sodium and chloride ions have been pumped out into the medulla region, or interstitial tissues in some textbooks it'll say, interstitial region. And the second mark then, what you say is, if sodium and chloride ions are pumped out of the ascending limb into this region, then they build up in this region. Sodium and chloride ions build up out here. And that causes a lowering of water potential in this region. So the buildup of sodium chloride ions causes a lowering of water potential. And then the third mark is, because you've got an area of low water potential out here, as the filtrate comes down the descending limb, water is pulled out of the filtrate, out into this region by osmosis. So it moves out of the filtrate where it's at a high concentration water, out into this region where you've got a very low water potential due to the sodium and chloride ions. Now those are the 3 marks, and essentially they're written very clearly there at the bottom of the page. So those are the 3 marks you get on essentially the 3 key points about the lip of Henle. However, you tend not to get the question asked just in that sense. It's simply don't ask you just to describe what's going on in the lip of Henle. They're more likely to ask you if you go to question, the most common question, most likely question, if you go to question 8, part C. And the kind of question they tend to ask you about is they ask you things about animals that live in different environments. And they tell you, for example, in this question, they ask you to compare a beaver and a kangaroo rat and with regards to their lip of Henle's. Beaver lives in a river in an environment where there's plenty of water, whereas the kangaroo rat is a little tiny mouse-like thing that lives in the desert where there's no water. And what you need to know is, the key point here is that animals that live in very dry climates where there's not much water, and i.e. animals that need to conserve water, animals that need to be able to reabsorb as much water from their filtrate, these animals have longer lips of Henle. So the kangaroo rat will have a longer lip of Henle, whereas the beaver will have a shorter lip of Henle. What's the advantage of a longer lip of Henle? Well, if you have a longer lip of Henle, gives more opportunity for more sodium and chloride ions to be pumped out. This lip of Henle extends way down further. More length of ascending limb for sodium and chloride ions to be pumped out. That creates an even lower water potential in this medulla region. It creates a much lower water potential in this medulla region. In fact, there's a question that I leave here. I leave in these notes, even though it's a very old question, but it illustrates this not too bad. Is question 3. It's an extremely old question, but basically question 3 shows you that if you measure the water potential as you move down the lip of Henle, you discover that the further down, the longer the lip of Henle, the lower goes the water potential in the medulla region. And you can see from that question, so you can imagine if the lip of Henle only extended down about halfway there, the water potential would be minus 800. But because of much longer lip of Henle, you've got a water potential of minus 1800. So having a longer lip of Henle. So here's the answer to this question about the, going back to question about the beaver, if I can find it. Question about the beaver and the kangaroo rat, which is, I have to find that question. Yeah, it's back to question 8 again. Let's see. The bottom of the page, given 3 marks there. Suggest and explain how the lip of Henle for the beaver would be expected to be different from that of the kangaroo rat. Well, I would just pick the kangaroo rat and just say the kangaroo rat's lip of Henle would be longer. And you'd say a longer lip of Henle allows more more sodium chloride ions to be pumped out of the ascending limb, creating a much lower water potential in the medullary, and that in turn pulls more water out of the descending limb. In other words, overall, a longer lip of Henle gets more water reabsorbed out of the filtrate in the lip of Henle, and if you get more water reabsorbed out of the filtrate, that means less water then makes its way into urine. So these kangaroo rats don't really produce any urine much at all. They produce a very small quantity of very concentrated urine because they need to retain and recover water. So that's the standard question, and you'll come across that question back and forward over the years. I like to ask about, with regards to the lip of Henle, I like to ask about the kangaroo rat or animals that live in wet environments. Okay, so that's the lip of Henle. You'll see that's well explained on page 12, at the top of right the way through page 12, and just the bottom page 12 says, why is the lip of Henle much longer than animals who live in a dry or arid climate? And there's about 4 points set out there. Now, we'll go back and look at more questions as we go along here. Finally, the collecting duct. And the term that describes what the collecting duct is osmoregulation. So if you're asked what the function of the collecting duct, you'd say it's osmoregulation. Basically, you refer to the collecting duct. Well, what has happened so far is you've got water reabsorbed in the proximal convoluted tubule. You get water reabsorbed in the lip of Henle, but the collecting duct acts as a sort of fine control. It's the last chance for the nephron to control the amount of water that's leaving in the urine. So it's kind of like a fine adjustment to make sure that the right amount of water is reabsorbed, and that happens in the collecting duct. Now, I said there it involves fine control of water reabsorption. And in the collecting duct, the reabsorption water is controlled by a hormone called Adh Antidiuretic Hormone, ADH, which controls the permeability of the walls of the collecting duct. It controls how easy water can move through the walls of the collecting duct, this hormone ADH. It's produced in the brain, in a place called the hypothalamus, and it's stored in the pituitary gland in the brain, and then it's released in appropriate amounts to travel in the bloodstream to the kidneys. So you have a mechanism then, there's a series of steps or series of stages in which you describe then about this ADH. And the key thing to know, the key thing to get into your head is how much ADH you are producing according to your intake of water. So for example, let's say you drank a whole litre of water, and then you have to follow that through and see if you drink a whole litre of water, that causes the water potential of your blood to rise. In other words, your blood gets very watery. That then is, as your blood then travels through your brain, you've got what are called detector or osmoreceptor cells, and they're present in the hypothalamus. Basically, they're like cells that dip into the blood essentially and measure its water potential, measure its soil potential. And if you drink a whole litre of water, then your blood becomes very watery, and in essence, you need to lose water from your body. In that circumstance, the osmoreceptor cells detect that the water potential of the blood is very high, and they produce less ADH. So you make less ADH if you drink a lot of water. If your blood is very water, your blood got a very high soil potential. Sorry, very high, very high soil potential. And vice versa, if you don't drink much water, and let's say you did a lot of exercise, a lot of running or something, your blood becomes quite concentrated, the soil potential of it goes very low, and that's detected then by the osmoreceptors in the brain, and they produce more ADH. And the ADH then travels in the bloodstream, and it affects the cells that line the collecting duct. Those cells that line the collecting duct are made more or less permeable. Now, the cells that line the collecting duct, how are these cells made more or less permeable? Well, they're made more or less permeable because they produce, or are producing those cells more or less aquaporins. Aquaporins are channel proteins that let water through. And if there's more ADH released, more ADH released by your hypothalamus, released from your pituitary gland into your bloodstream, the effect of that ADH is to cause those cells lining the collecting duct to produce more aquaproteins, and those aquaproteins are basically channel proteins. So if you have the wall, say these are the cells that are lining the walls of the collecting duct, and let's say the filter is coming down here, and these cells produce more aquaporins. And basically, they're just essentially like little pores, more proteins that have pores, and that effectively allows the water then to escape from the filtrate. So the filtrate's in here. If you get more aquaporins produced, then more water can escape through the walls of the cells that line the collecting duct. Now, on occasions then, they've asked you just to describe the mechanism by which ADH controls the reabsorption of water at the collecting duct. In fact, I use a question there. You'll see a question in the question booklet that asks you to describe. I think it's somewhere in the question booklet there, maybe not. Yep. Yeah, question 10, which is an essay question. And the second part of it says, describe and explain the process of ultrafiltration and reabsorption. Sorry, second part of it, part B, it says, using osmoregulation as an example, explain the term homeostasis and outline the essential components of homeostatic mechanisms. Now, see, for that, there were 6 marks for that. Basically, they're asking you to, first of all, say what is homeostasis, which is straightforward enough. And then they're asking you to discuss how this ADH system works. And essentially, it's referred to as, well, it's a homeostatic mechanism. And essentially, any homeostatic mechanism, any mechanism that's going to keep something constant, needs some way of measuring how much of the substance is present. In this case, you need a mechanism or a detector. In this case, in the detector, you'll see there, it says, in the first point, looking at the mark scheme. By the way, the mark scheme for that essay is tagged onto the end of the notes there, so I'm looking directly at it. It says, the osmoreceptors are the sensors in the hypothalamus that allows the blood water solute potential to be monitored. So first of all, you need a detector. If you're going to keep something constant in the body, you need some way of detecting whether or not it's moving, whether it is near its normal level or it's moved away. So you have the osmoreceptors, and then basically the idea is that you have a set or normal level for the blood water potential, and if that deviates away from that, then the corrective mechanism, in this case the ADH mechanism, comes into place. So you have, like in the body, a normal solute potential of blood, and then if the solute potential of the blood goes too high, based on, you can see on page I deal with that on, the solute potential goes too high, which is on page 14 minutes, then that's detected by the osmoreceptor cells, and that produces then more ADH. Sorry, that produces less ADH, so the solute potential goes high. That means it goes more towards 0, so you get less ADH produced, and that less ADH then doesn't make the walls of the collecting ducts too permeable, collecting duct cells too permeable, and more water can continue out in the urine. Whereas if your blood water potential decreases, then that's detected by the osmoreceptor cells, and they then produce more ADH, and that increased ADH makes the walls of the collecting ducts more permeable. So that's set out on page 15 minutes there. The mechanism is said to be a negative feedback mechanism. That term simply means that the mechanism responds to the extent of the deviation away from the norm. So if the blood water potential is a long way away from the norm, and the mechanism responds in a rapid or in a large fashion, as it were, but as the blood water potential comes back closer and closer to the norm, the mechanism is effectively switched off. So it's reduced and switched off. So the response of the ADH mechanism is big if there's a big deviation, but as the deviation reduces, as the blood water potential goes back towards the normal, there's less and less response, less and less response in the ADH system. The way I've said it there is that as blood water potential rises towards the normal level, for example, less and less ADH is released, i.e. a return towards the normal starts to switch off the mechanism. Okay, so that's dealt with most of the areas of this topic. Now I want to just zero in on a few questions. Question 1A, that's the identification of different parts, that's okay. I want you to make reference to, I want you to look at part 1B, and also the same question is, question 1B there is a fairly recent question, but the same question has been asked over the years in a slightly different format. So if you go to question 7 as well, so I want you to have question 1B and question 7 both open. And I've dealt with the issues on that question, those questions on page 16 of the notes. Page 16 of the notes. So what do we see in question 1B and question 7? Well, what are those two questions looking at? You'll see that both of them are looking at concentrations of substances in the different regions of the kidney. So I'm looking at 1B here. They're looking at 4 different regions: the PCT, proximal convoluted tubule, lip of Henle, distal convoluted tubule, and collecting duct. And they're looking at different substances, looking at urea, chloride, sodium, and glucose. And the same, if you look at the question 7, it's a very similar question, except instead of having a graph, it's just got a bunch of figures. But essentially, it's the same. It's measuring the content of the filtrate, and it's measuring it for protein, glucose, sodium, and urea, and it's measuring it at different places. So it's got the amount of these substances at different places. So I'm just looking at question 7 there. Just for example, the amount of protein, and number 1, it says plasma in the afferent arteriole. So you see there's 80 grams per dm cubed, dm to the minus, that's per liter. So it's saying that the blood in the blood that's arriving at the kidney contains 80 grams per liter of protein. 80 grams per liter, that's quite a lot of protein. And you can see the blood contains 1.2 grams of glucose per liter. That's not very much. A whole liter of blood, 1.2 grams of glucose, and 34 grams of sodium ions and so much urea and so on and so forth. Okay, and then you can see that measured the amount of those substances that are present then, how much of those substances go through the filter. I'm looking at question 7 again, that would be filtered in the bonus capsule. And then they're looking at the end of the proximal convoluted tubule, the bottom of the lip of Henle, etc., etc. And they're seeing how much of the substance. But I think question 1 part B is the most recent question, so I'm going to look at it quickly. And I'm going to look at glucose first of all, and you can see what you see for the glucose is the concentration of glucose decreases very rapidly in the proximal convoluted tubule. And that's because all the glucose gets reabsorbed. So any glucose that's in the filtrate is reabsorbed in the proximal convoluted. It's all reabsorbed in the proximal convoluted tubule. If you look then, we'll move on to the lip of Henle. Now, the most noticeable thing about the lip of Henle, if you can see that I've kind of drawn a little lip of Henle here, there's the descending limb, there's the ascending limb. And you can see in the descending limb, the concentration of sodium and chloride ions go up. And then in the ascending limb, the concentration of sodium and chloride ions come down. Well, let's deal with that. Let's deal with the ascending limb first of all. Why does the concentration of sodium and chloride ions in the ascending limb of in the lip of Henle decrease? And the answer is because sodium and chloride ions are being actively pumped out in the ascending limb. So their concentration in the filtrate decreases. Yeah? And then why does the concentration of sodium and chloride ions go up in the descending limb? Because the water is being extracted from the descending limb. If you extract the water and the sodium chloride ions aren't extracted in the descending limb, then the concentration of sodium chloride ions go up. Does that make sense? You'll notice for urea, the concentration of urea just keeps going up and up and up the whole way along. And that's simply because no urea gets reabsorbed. Never gets reabsorbed. But because you're getting water reabsorbed in the proximal convoluted tubule lip of hell, etc., etc. Because you're getting water all the time reabsorbed, then the concentration of urea increases because the urea is now dissolved in less water in the filter. And of course, you can see further along here, sodium and chloride ions go up too. More water being reabsorbed. You can see in the collecting duct, the concentrations go up because water gets reabsorbed in the collecting duct. So essentially, the most marks there are for, you'll see, well, first of all, part one says describe the trend shown for glucose in the proximal convoluted tubule. Concentration of glucose declines rapidly. Declines, not maybe declines dramatically is a better. And you'd say because water is being reabsorbed in the proximal convoluted tubule. And then explain the changes in the concentration of sodium and chloride ions along the lip of Henle. 4 marks. The changes in the concentration of sodium and chloride ions. And you would say in the descending limb of the lip of Henle, water is being reabsorbed from the filtrate, and therefore the concentration of sodium ions must increase because water is being reabsorbed. And then you'd say in the ascending limb, sodium and chloride ions are being actively transported out of the filtrate, they're being reabsorbed out of the filtrate, and because you've removed sodium and chloride ions from the filtrate, their concentration is decreasing in the ascending limb. Yeah, that's it. Okay, so I'm just scanning across questions here. Looking, I'm leaving out some bits because it's just I won't be able to cover everything. Question 2 is quite a recent question. I think it's 2018's question. Explain the term ultrafiltration, the filtering of small molecules out of the blood plasma across the filter in the kidney at high pressure. You did the high pressure thing. 2 ways in which the glomerulus is adapted. Afferent arteriole is wider than the efferent arterioles. That creates high pressure. And you also say the glomerulus has, you could say it's convoluted. Glomerulus is the winding of the blood vessels wrapped around each other, and a knot creates high pressure. Or you could say about the blood, the pressure of the blood reaching the glomerulus. Pressure of the blood coming to the glomerulus is very high because of the short distance to the heart. Name one substance found in the blood in the afferent arteriole that will not be normally found in region A. Something that's in the afferent arteriole. So afferent arteriole is the one coming. And region A is the something that wouldn't get into region A. You could say large proteins, because large proteins don't get through the filter. Good. The rest of that question then was really quite difficult. The relationship between the toxicity of an excretory substance and the volume required to remove it from the body. As the toxicity of the substance increases, the volume of water increases. Is that right? The two are proportional. And suggest a reason for this. If you more toxic substance, you need to dissolve it in more water to reduce its concentration, to reduce its danger to the body. So higher volumes of water needed to dissolve more toxic substances. One other advantage of making a less toxic excretory waste product, well, the other advantage is you don't need to put so much water. You don't need so much water to dissolve a less toxic waste product. And therefore, the body doesn't need to lose as much water. Less water is required for its removal, so more water then gets to stay in the body. I'll leave you to look at the rest of that question. I just won't be able to get through all the questions. Look at them, look at everything. So essentially just come back to question 7 with the table. Essentially question 7 is like question 1b, and you can see the most difficult part is part B, where you get 4 marks for the sodium and chloride ions in the Nephron. So essentially that's the difficult part. I like part E. Part E of that question then is asking about reabsorption in the proximal convoluted tubule at a lower temperature. And basically you'd say lower temperature means a lower rate of respiration. It means less energy released for the active transport proteins in the lip of Henle, in the walls of the lip of Henle, and therefore the lower the active transport, the less glucose gets reabsorbed then, because the active transport proteins are not working at full steam, as it were, and so therefore some glucose ends up some glucose doesn't get reabsorbed, so you end up with glucose in the urine. Scanning on across the questions there, all the usual all the usual types of questions. So, yeah, I'm just going to stop there now. So, I think we covered most of it, covered nearly all of it. Good. Right, the best thing for you to do then is to do all the questions and look at all the write out through all the answers, and that should help. Okay, hopefully this has been helpful. Thank you very much. Goodbye.